Okay, so to start off with, we're gonna start to talk about glycolysis. So for glycolysis, we need glucose. So for glucose, which is gonna be 6 carbon, so you see 1, 2, 3, 4, 5, 6 right there, and we're gonna need 2 ATP molecules, and then we're gonna need 2 NAD plus molecules. So as a result of the glycolysis, we're gonna be making 4 ATPs. Remember, we put in 2 right here, and we gained 4. So basically, we made 2 because 4 minus 2 is 2. And then we made pyruvate. We made 2 of them actually. So each pyruvate is 3 carbon, and then remember the glucose was 6. So this is 3, and you're gonna have 2 of them. So that's 3 times 2 is 6 carbon, right? And then we're going to be making two NADH molecules. So you see, I drew a dead battery for the NAD plus. That means it's like lower charged. And then NADH is like completely like charged, like a full phone battery or something like that. Yeah. And glycolysis, you need to remember it occurs in the cytoplasm, and it occurs with or without oxygen. And that's about it for glycolysis. And next, we're going to be moving on to prep steps. So we're going to talk about that. And I forgot to add, we need the substrate level phosphorylation is basically what it means is like it's you break a molecule and then you gain energy. Like that's basically like gain energy by like ATP, right? So that's what substrate level phosphorylation is. All right. Okay, so the second step is going to be prep step or half step or intermediate step. Okay, so for this one, you remember last time we made 2 pyruvate from glycolysis. So that from here goes to here. So we're gonna have 2 pyruvate from the glycolysis step as a product of the glycolysis. We're gonna be using that. And so remember, the pyruvate is each of them is 3 carbon, and since there are 2 of them, they're gonna be 6, right? Okay. And then you're gonna have 2 coenzymes A, so I just abbreviated it right here. And then the 2 coenzymes A, they're gonna attach to like the 2 carbon molecule, but wait, there aren't 2 carbon, it's 3, right? So basically when the coenzyme A attaches to the pyruvate, the one of the carbon, like you see like there are 3 of them, when this attaches to like this, one of them goes off, it falls off. And then the carbon that falls off, it's basically, it's like, I don't want to say exerted, but it's like gone as like CO2, like carbon dioxide, right? And next we have 2 NAD plus molecules, so they're like, as I said, dead, like lower charged, right? And as a product of, so this arrow is showing like what are going to be like the products of the prep steps. So we're going to be seeing, we're going to be having 2 acetyl coenzyme A molecules, and they're going to be 2 carbons in each of them. So we're going to have 4. All right. And what basically like what this means is like it's going to be acetyl means like it's the 2 carbon part, and then coenzyme A right there, right? It's like 2 carbon. That's what it basically means. That acetyl. That's what it means. And there are 2 of them, obviously. So that would 2. And then you're gonna have 2 CO2 as a product, and I told you how. And then you have 2 NADH. The 2 NADH, see, it's full battery, green, fully charged. That's what you get. Okay, the next step is gonna be the citric acid cycle. Wait, before I go, I just want to make sure I said when this occurs, we're moving into the mitochondria. So the glycolysis, it occurred in the cytoplasm. Now when this step starts, we're moving into the mitochondria, right? And then for this step, oxygen must be present for this step, right? So next step, we're going to be moving into the citric acid cycle, and then for that step, we're going to be moving into the matrix of the mitochondria. All right. Okay, so for now, we're in the citric acid cycle, also known as Krebs cycle. Okay, so it's a cycle because it's a regenerated process. So basically, what that means is like the products, like the thing you start with, when this ends, you're like basically right there where you started at, kind of. Okay. So we're going to be using the two acetyl coenzymes A, and remember that came from right here, which is in the prep steps, right? That was from here. Use those, and that comes right here. So for that, you're going to be, the two acetyl coenzymes A, you're going to be reacting with oxaloacetate. So the acetyl coenzyme A was 2 carbon, then oxaloacetate is 4 carbon. See, 1, 2, 3, 4. All right. So when you see like all this process occurs, this whole circle, the cycle, it occurs, you're gonna be making 2 carbon dioxides. So when I say two, I'm meaning like for one cycle, and and this process occurs 2 times. So in like when this goes one time, you make 2 carbon dioxide. So if it will go 2 times, you would make 4, right? Basically, yeah. So then next, there's going to be 1 ATP for one cycle. That means 2 ATPs for 2 cycles. And then 3 NADH for one cycle. And then 6 NADH for 2 cycles. And then 1 FADH for 1 FADH2 for one cycle. And then 2 FADH2 for 2 cycles, right? And for this one, this occurs in the matrix of the mitochondria. So last time we were just moving into the mitochondria, and now we're inside the matrix. And for this one, it's going to be a substrate level phosphorylation as well, because we're making ATP. So just to go over like what we're making in this step is going to be like the total. So we're going to be making 2 FADH. So it was 1 for the one cycle, and then times 2. 1 times 2 is 2. And then 6 NADH, so 3 times 2, it's going to be 6. So, and then we're going to have 2 carbon dioxides, so 2 CO2s. And then 2 times 2 is 4. And then 1 ATP for one cycle, 2 ATP for 2, so 1 times 2 is 2. All right. Okay, so next step, we're going to be talking about, wait, before I go again. So oxaloacetate, for this thing right here, oxaloacetate, it's a regenerated molecule because it's used over and over again. I just wanted to make sure I said that. Okay, next step is going to be the electron transport chain, also known as ETC. All right, so we'll talk about that next. Okay, so, and in this step, we're going to be talking about the electron transport chain, ETC. All right, so let's make sure we get to the diagram right here. All right. Okay. So basically, what's going on over here, and it's a lot of things, words, molecules moving around. Okay, let's break this down. Okay, so right here, we're gonna have a hydrogen ion, basically a proton, build up in the matrix. So right here, you see we have the matrix, and so this is like a phospholipid bilayer between the intermembrane space and the matrix. This is a phospholipid bilayer. So you see I drew the phospholipids right here, and it's a bilayer since there are 2 of them, right? And these proton pumps, and this proton pump, and then ATP synthase, which is an enzyme, it's going to be embedded inside the inner membrane. So embedded inside the phospholipid bilayer, right? Right here. Okay, so I was talking about the hydrogen ions, the protons. So you see, and remember, the more hydrogen ions there are, the more acidic it gets. So like, since it's more acidic, it can't be more acidic. That's a problem, right? So what's gonna happen is the electrons, which came from the breaking of the glucose molecule. Remember, we broke the glucose from the glycolysis step right here. So those electrons, which is like free energy, so those are gonna come into play, and then these are just gonna go inside the proton pump, and then the proton pump, it's gonna, what it's gonna do, it's gonna open the doors, like the doors, basically. It's not a door like calling a door, like this is a door maybe. So what this is gonna happen is, like, the electrons, when they go in, they're gonna open it. So when it opens it, so what's gonna happen is the hydrogen ions, the proton, remember, it's a proton pump, so hydrogen ion, which is a proton, it's gonna go across this thing, the proton pump, allowing the protons to go across. So the protons go from the matrix to the intermembrane space, right? Okay. So what's gonna happen is next it's so like the electrons, what I was talking about, like the electrons, they don't stay in one space. They're not gonna stay like in one proton pump for very long because it's gonna like destroy it. So it's gonna move. So it's gonna move, and you see I put the CTYC, so that's the cytochrome C. So what its job is, basically it makes sure like the electrons stay on their path. So electrons, they're moving from one proton pump to the other proton pump, and then the cytochrome C makes sure that it stays in, like it stays on its path. The electrons stays on its path to the other proton. So when this electron, sorry, I meant electron. The electron stays on its path, and then electron from proton pump 1 goes to proton pump 2, and then so like the proton pump on the first one, it's not gonna stay open, so like it's gonna go, like as soon as the electron leaves, it's gonna close. So it's not like if it came once it's gonna stay open, no. So it's gonna go from here to here. So when it comes over here, so the door for this one, for this proton pump, is gonna open, right? And then the hydrogen ions, the protons from here go up there. So you see, basically what's happening is, so you'd be thinking like, okay, so we had protons over here and hydrogen, like hydrogen ions on this side in the matrix, that was more acidic. Now you basically moved them to the other side into the intermembrane space, so isn't that more acidic? Well, yeah, technically. But what's gonna happen is this ATP synthase right here, the enzyme ATP synthase, so that's like the really cool part. So what's gonna happen is in the ATP synthase, the hydrogen ions or the protons, they can only move in one direction. So it can only go this way, right? It cannot go like this, up and down, up and down. It can just only go down into the matrix, right? So what's gonna happen is when the protons that came from here went up there and they made like a bunch of protons over here, they're gonna move from here to here when it's gonna go from here to here. Well, not so quickly. So when it's going down to the ATP synthase, the protons, when they're going down, so what's gonna happen is like, so like basically this thing is like a windmill. Like think of it as like a proton mill, right? So what's gonna happen is, like, you know, like on a windmill there's like the blades. So as soon as the wind hits, the blade spins, right? So what's gonna happen is, like, as these hydrogen ions or like protons, they when they hit like the blades, these blades right here, when they hit them, the proton hit the blades. So what's gonna happen is, like, it's gonna spin, and when it spins, it's gonna when the blade spins, the blades are gonna allow ADP, and then the I don't know how to say that, the pi it's gonna attach onto it. So when they attach onto it, and it's gonna keep spinning. So when it spins, what it's gonna do, it's gonna throw, like physically throw the phosphates last what's it called, the electron cloud, to the pi. That's what I'm gonna call it, pi. And that's how, when it spins, when the last electron cloud, like the last electron cloud is thrown from here to there, so that's when, like, the ATP is going to be made, and then that's where the most of the ATP is going to be made. So, and you see, like, so you'll be thinking, okay, so you sent hydrogen ions from here because it was too acidic, you made acidic over there, and then you send them back down, and then you have basically keep moving around, and you're gonna have acidity all the way around, right? Well, not so quickly. So what's gonna happen, remember we had electrons over here, right? So these electrons are gonna come in over here, and then what's gonna happen is the oxygen, it's gonna come and pick up the electrons. And then oxygen is a diatomic molecule, right? So there's going to be hydrogen ions right here, the protons. It's going to pick up those and those. It's going to make what do you think it's gonna make? It's gonna make H2O, water, right? So it's gonna result in water. And so basically, I just want to make sure you see this. So you're going to be making 34 ATP molecules from the electron transport chain, which is this step right here, which we just talked about, this whole thing. And then you're going to be making 34 from there, and then you're going to be making about 4 ATP from like the glycolysis step and like all that, the prep steps, not the prep step, but like the citric acid cycle, all from like the other steps. You're going to be making 4 ATP. And remember how I said about the H2O, how it's going to be making water? It's called chemiosmosis. So chemi means like it's a chemical process, right? So chemiosmosis because it's making water. So osmosis is water, and then it's gonna chemi because it's making it from like a chemical process, right? So chemiosmosis. That's what that's gonna result. And then at last, before we finish, I just want to talk about fermentation. My bad, I forgot to talk about something else again. Okay, so I forgot to talk about oxidative phosphorylation. So what basically oxidative phosphorylation is, it's the like oxidative phosphorylation, like how we talked about like the substrate phosphorylation right here. It's gonna be basically oxidative. So oxidate means to like lose electrons, right? So when electrons are lost, so oxidative phosphorylation basically means the making of ATP through the loss of electrons. Okay, that's what it means. So basically, it's like the passive. Let me say, like, choose to summarize the electron transport chain. I just want to say it's like the passive movements of like hydrogen ions from high to low concentration, and then across the inner membrane right here, and then from the matrix to the intermembrane right there, and back through the ATP synthase. And this is called chemiosmosis because, as I told you, it's going to be making water. And through the movement of the hydrogen ions, as I told you, they grab onto the blades, it spins, and it's going to be making ATP, right? So as a product of the electron transport chain, you're going to be making 34 ATP and water. So water is like a byproduct, right? And NAD plus is a coenzyme of oxidation and reduction, and they move electrons from one place to another. So it's like basically a carrier. It's like transporter, right? It's like a bus driver or a bus. Okay, and then cytochromes, as I told about right here, I talked about cytochromes. The cytochromes are proteins embedded within the proton pumps, like not within, like with the proton pumps that help the passing of electrons. So as I told you, electrons passing from here to here, so cytochrome C, it's going to make sure it stays on its path, and it's going to move from here to that proton pump. All right, I forgot to talk about that. Okay, next we're going to talk about fermentation at last. Okay, so at last we're going to be talking about fermentation. Okay, fermentation occurs if no oxygen is present and then pyruvate is still made, but fermentation occurs within the cytoplasm. Okay, so for fermentation, it doesn't, like, oxygen is not needed, so it's like anaerobic, and all the other ones were aerobic. So anaerobic is like doesn't require oxygen, and then aerobic oxygen is required. What else? Okay, so basically you might remember, like, you recall this diagram from our notes, where we had glucose, 2 ATP. This might look like the glycolysis, but it is. And then for this one, we needed to have oxygen, but like for this one, this, like, you don't need to have oxygen for like glycolysis, but you know what I mean. Okay, so glucose is so like you see glucose over here. Glucose is incompletely metabolized to lactate or to carbon dioxide and alcohol. So like glucose, blah 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 blah, then you make um, carbon dioxide and alcohol only in plants. Okay, I skipped a lot of steps, but let me show you. So fermentation basically is an anaerobic process. And then the advantage of fermentation is, let's say like you need like a quick burst of energy, like you're gonna get that from fermentation. That's an advantage. There are way more disadvantages than advantages. So some of the disadvantages are gonna be, it's the lactate and alcohol it produces. It's gonna be toxic to the cells. And then the lactate changes pH and causes the muscles to fatigue, which is known as like the O2 debt. This is why like you breathe like really fast after you like run out of breath. And then yeast, they die from the alcohol they produce, so that's like bad. Okay, so let me just like explain this real quick. So you're having two ATP molecules, same as glycolysis, going through making two NADH, four ATP, basically two, but we're gonna say four. And then you're gonna have two pyruvates, three carbon each. And then after that, this is where fermentation occurs. So fermentation, um, you're gonna be making two lactates, which is gonna be only in animals. Um, and you're going to be making carbon dioxide and alcohols in plants. Okay, and this will continue with the use of glucose and break down into two lactate or two alcohols and two carbon dioxide each time, only net gaining basically two ATP molecules. So it's like basically making two. All you do is just make two out of it, and then you can't continue for like a very long time. It's going to be for a very, very short time.